Hello everyone and welcome back at another video for the Utrecht University course Introduction to Bioinformatics. This is another video for topic number 2, metagenomics, and I'm going to take you through exercise number m05, backmapping. So let's have a look at the actual exercise. One of the lessons that you will learn in this exercise is to read the manual. There's a lot of instructions at the beginning. I'm gonna skip these for now. I'm assuming you've read all of these. So the first actual step that you are doing yourself is to index the assembly with BWA index. So the assembly here, the metagenome assembly, is synonymous in this case for the scaffolds.fasta file we've been looking at earlier. So let's just double check that we have that already. Assembly scaffolds.fasta, yes, it is there. And now let's have a look at what we can do with this BWA index. So BWA is an aligner. It's basically a big DNA search machine, if you will. But before we can search for something, like in an old school book, you could make an index at the very end of the book where there's some search words to find stuff in the book easier or quicker. Same is true for genomes in a way, I guess. And we can do this with BWA index. We make this search index of our FASTA file. So let's see what BWA tells us. So sometimes you could just type a command. Let's just type BWA just for fun. Sometimes when you do this, type a command without an argument, it will give you an error. Sometimes it will give you the manual. In bioinformatics, I think often it's the case that the program will actually give you information. But sometimes you will need to type something like -h, the option for help. In this case, BWA doesn't recognize this. Sometimes you type --help. BWA also doesn't recognize this. So let's start here. BWA. There's all these options, and in this case we want to use the index option, BWA index, and the first line, that's always a good one to look at, is the usage line. So how do you use this command? So you type, like we did, BWA index, then there's options, which of course are optional, and then there's the input FASTA. So I type the command, I use tab to autocomplete, so I can't make any mistakes. And I'm not using any options now. I hit return and I wait a bit. Making an index like this can take some time. It needs to go through the entire assembly and construct somehow a set of files that make it easier for BWA to search through this assembly. So this can take a bit. Once this is done, we'll check what files come running out of this, and then we'll start the actual backmapping process. Yeah, so I'm gonna pause this video just for a bit while it is indexing. See you in a bit. Okay, so the indexing is done. We can see in the output that it gives us all kinds of information that we likely will not understand. So let's see what actually happened. We'll have a look in the directory where the original scaffolds file is, so that's data/assembly, and see what's there. So before there was only one file, scaffolds.fasta. Now there's actually multiple files, and they all have the same base file name, as we call this, but different extensions added to this. So what these different files are is a course on its own, so I'm not going to talk about that, but just know that all of these files together are the index that we made. So these files combined are the result of the step we just successfully performed. And if later BWA asks for the base of the index, it asks for the part of the file name that's shared among all of these files. So that in this case is very easy, because it would just be the name to scaffolds, the path to scaffolds.fasta. Okay, so that's done. Next thing is we're gonna run BWA mem for backmapping. So here's a couple of steps, and if you got confused at this point, that's okay. Everyone does, always, every year when we do this course. but it is a bit of a challenge, so I like to keep it in for you. Let's see, let's follow the steps. So we are going to make a new directory for the SumTools view result. And remember, SumTools view is this program that we use to store the BUM files, which are the result of our backmapping. Give it a useful name. I'm just going to follow my own instructions. I'm going to make a directory, mkdir, make dir, data/mapped. Hit return. Done. I've now made a new directory, an empty one, in which I will store these results. Next step, I made a loop for you to play with. So a loop you practice with in the Jupyter and Bash primer that we made available before the start of the course. Now you will need all your knowledge of the primer, what we did this morning, and the lecture, and we combine this into one. Let's first remember how a loop works. So I made an array. An array is a list of variables, so to say, and the array is called samples. That's how you'll often find your stuff named when you work with biology. And if I just execute this cell, it does a thing for each of my names in the array. So I could add something to this. Let's put coffee, because I really like coffee. And there we go. It's now part of the array. I could remove coffee, and inside this loop I could say echo $i, the variable dot fastq. Could be a thing, and it will print this. So just remember how a loop works, and if it's unclear, try to go back to either the primer or just play a bit with this. I'm gonna leave that as it is now. Let's see. Don't forget to read the manual page of both, and in both I refer to BWA and SumTools. So BWA-mem. Let's have a look at the manual page. It tells me how to use it. So BWA is the mapping algorithm. It takes short reads and aligns these to the metagenome assembly that you were given using the index that we just made. So BWA-mem, you can put in options. You don't have to, but you can. The index base, so remember that's what we just made. Then required also is one input file .fq, which is short for fastq. And optional, a second in file. We do have that second input file, so we definitely want to use that. There's all kinds of options that we can use. So let's say you're after this course starting your own research project and you want to process your samples fast. You may want to use more threads, use more processors inside your computer. Today we do not want to do that. We want to be social to the other people in the course. SumTools view, let's have a look at the manual page of this. SumTools view, then there's options, there's an input.bum, and there's some other optional stuff that we can put. Some of the options are to put out a BUM file, which I think is default. There's all kinds of compression stuff that you can edit, but you don't have to. These things are optional. So this takes a lot of space, and since I know already what it means, I'm gonna remove these to make it a bit easier. BWA-mem does not require any options. For SumTools view, look for the option. Okay, we're gonna do that. Should look somewhat like this. BWA-mem. Then there's some argument. Of course, here I hint at the index that you made. That's what we just saw on the usage line. Then there's the fastq file that you put in. We of course put in the second fastq file as well, since we have this data and we want to have the optimal benefit of this. We use the pipe character as shown here and move the data straight to SumTools view to make it a BUM file, and we can put here whatever. So let's have a look. Yeah, so I put in a little bit of stuff here for you. I put in actually a lot of stuff here for you. So that's cheating. let's do it from the basics. bwamem. If you forgot, and I am forgetful, so let's get the manual page. Am I still here? bwamem. Let's have the manual page here and see how it works. bwamem, and there's the index, so that was easy. The index is the same for all of the different samples we have. Data scaffolds, no, assembly / scaffolds.fasta. Remember, we put in the base file name, so index base is what it says here. So we use the part that's shared by all of them. Now comes the tricky part. We want to put in FASTQ file number 1 and FASTQ file number 2. So we want to do this for these 6 different samples that we have, and for each of these samples create a different output file. So how would it look like? If you find variables hard, and most people find that in the beginning, let's do it first without a variable. How do we want this to work in the end? So what goes in is data / reads. That's where our raw data is. And let's say we take sample E1 first. There we go. And we do the same for data / reads. Come on, reads. e1.r2.fastq.gz, because these files are still gzipped, so to say. So what I have here now looks exactly like the manual here. Great, happy with that. So the next thing, let's look at our step plan here. bwamem argument blah 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 blah. We've done this. Pipe SumTools view. Okay, we can do that. Do a pipe SumTools view. And let's have a look again at the manual of SumTools, just because we are forgetful. SumTools view, then there's options. And forget the options now, and then there's the input. So now what the manual doesn't say is that if you give SumTools the input file in a pipe, you don't need to specify an input file. It doesn't make sense, you already gave it an input. So actually the input here is not applicable in our case. Let's see what else we have. We want to make a BAM file. So I type -b because that's what it says in the manual. I think if you forgot this, it's fine. I think BAM is by default the setting. And it would be nice to have an output file name. We want to know where our stuff is stored. So you could type -o like this. We made this folder, remember, data / was it mapped? And in this case I want to call it /e1.bam. And then what will happen is we'll take the assembly of all samples together, the reads of sample E1 forward and reverse. It will map these and then store these in a good and efficient format in a file called e1.bam. So every time we take 2 files and we turn this kind of into 1 file. Now remember, if we execute this loop, it will do 6 times the exact same thing. That's not what we want. We wanted to do 6 times the same thing but for different files. So how would we do that? We want to substitute this E1 for the samples, for the different samples. So we can type $i in here, and in the first loop it will put in $e1.r1.fastq.gz, etc. And in the second one it will do E2, the third one E3, etc. I substitute all the instances of E1 by a $i, which is the variable inside the loop, and I am done. I will remove this stuff here. Again, this will take quite a while to run. Let's hit Ctrl return and see what happens. Looking good. There's still this green dot on the side. If you are hungry and you fancy lunch or a coffee break, now will be a good time. This is gonna take a bit. Yeah, so let's take a final second. This is a long video. Let's take a final second to think about what we're actually doing. So there's a whole technical challenge, but what's actually happening? So remember that these different samples we have, E1, E2, E3, P1, P2, P3, these were originally one biological sample that went into a proverbial blender and DNA was extracted. In this sequencing process we then got two separate files, so I gave you 12 files to work with, 12 sequencing files originally taken from 6 samples. These were assembled into a metagenome, basically meaning you take millions of very tiny pieces and make from them thousands of slightly longer pieces. And we now want to know, of all these slightly longer pieces, which I call scaffolds, how much of the original sequencing data actually maps back onto these scaffolds. So why do I want to do that? For all the scaffolds, all the pieces of DNA that have a similar abundance of DNA mapping on there, I can then conclude that these were more abundant in the sample that they were sequenced in. So this gives me a biological signal. A lot of technical steps, but all towards an end, a biological signal, information about our data. So out of here come, instead of the 12 files we had earlier, come 6 files, and in the next step we'll summarize this information even more. But that's for the next video. See you in the next.